You know, it's pretty wild to think about the sheer architectural genius behind every living creature. We start as just this microscopic ball of cells, right? And somehow that little sphere transforms into something as complex and recognizable as, well, as you or me. Today, we're diving deep into that really astonishing transformation.
Right.
It's a pivotal moment in development known as gastrulation.
Exactly.
Gastrulation is when that simple hollow ball of cells, the blastula, rearranges itself into a multi-layered embryo, the gastrula.
Mm.
It's this breathtaking ballet of cell movements and selective adhesion that truly lays the foundation for everything that comes next.
It really does.
And to understand this intricate dance, we'll lean on what biologists call fate maps, which are basically the embryo's earliest blueprints.
That's the key.
So our mission today is to peek behind this biological curtain, understand the how and why of these early changes, and explore the incredibly diverse ways different creatures tackle this fundamental step of life.
Yeah, and what's truly fascinating about gastrulation, really, is that to even begin to predict it, we first need to read these fate maps.
Right, the blueprints.
Exactly. Think of them as like pre-visualization charts. They show us precisely what each region of an early embryo is destined to become down the line.
Mm.
It's almost like having a detailed diagram of a building before the first brick is even laid, tracing where every single component is going to end up.
So how did they figure these out? I mean, you can't exactly ask the cells.
Huh. No, not quite. The technique that really gave us these insights was pioneered by a scientist named Fock. He developed this clever method of marking specific areas on early embryos using things like tiny agar chips soaked in vital stains, safe stains, of course.
Okay.
And by carefully watching where these marks migrated and what tissues they eventually formed in older embryos, Vogt could meticulously chart the fate of each region. So when you see terms like prospective epidermis or presumptive neural plate on a map, it's a direct reference to what that specific patch of cells will become. It's predictive. And the larger significance of these maps is just profound. They're not just static diagrams, they're dynamic tools. They let us track the incredible journey of these cells and really understand the precise orchestrated choreography of movements during gastrulation.
That's fascinating. It's like a GPS for development. So with that blueprint tool in hand, which creatures kind of jump out as sharing a surprisingly similar basic plan, even if they end up looking totally different?
Well, that's a great question. The tiny sort of fish-like marine animal Amphioxus and amphibians, like your everyday frog, they immediately come to mind.
Really? Amphioxus and frogs?
Yeah. Despite being really distinct organisms, their early developmental blueprints, their fate maps, show these striking resemblances. In both, you see an animal region in the early embryo that's fated to become the prospective epidermis, the outer skin, and the neural plate, which forms the nervous system. Together, these make up the ectoderm, the outermost layer of the gastrula.
Okay, the outer layer. Makes sense.
Then, if you move inwards a bit, the middle zone, sometimes called the marginal zone of the early embryo, is destined to form the mesoderm. That's the gastrula's middle layer.
And the mesodome is important, right?
Oh, incredibly important. It includes the prospective notochord. That's a crucial supporting rod that forms beneath the neural tube, giving structural support early on.
Okay.
And also the non-notochordal mesoderm. And that is incredibly versatile. It develops into all sorts of things like our limbs, heart, muscles, kidneys, gonads. The list goes on.
Wow, quite a lot comes from the middle.
It really does. And finally, the vaginal region, usually the bottom part in these diagrams in both Amphioxus and amphibians, it consistently gives rise to the endoderm.
The innermost layer.
Exactly. The innermost embryonic layer, which will form the gut and all its vital associated organs like the liver and pancreas. It's this remarkably conserved, three-part plan.
So a clear three-layer system. Ectoderm, mesoderm, endoderm. Pretty straightforward. But you mentioned Vogt's foundational work earlier. Is our understanding of these maps like totally locked in? Or are there still surprises?
That's an excellent point, and it really highlights how science works, doesn't it? It's dynamic.
Yeah.
While Vogt's work absolutely laid the groundwork, showing us the core principles, our understanding is constantly being refined.
Yeah.
Like more recent work by Keller and his colleagues,
Okay.
specifically studying the South African clawed toad, Xenopus, a really common lab amphibian. They demonstrated that structures like the prospective notochord and some other mesoderm components are actually located in deeper positions within a late blastula.
Deeper. So not just on the surface map.
Exactly.
Yeah.
Not just on the surface, as Vogt had initially depicted. And this isn't just some minor correction. It's a powerful insight into the hidden depths and the precise movements happening even before gastrulation fully kicks in.
Hmm.
It shows that even our foundational blueprints are always open to, well, deeper revelation. There's more going on underneath.
That's incredible that even the blueprints have hidden layers. Okay, so we've got this three-layer plan, more or less, for Amphioxus and amphibians. But what about something totally different, like a bird with that enormous yolk? I mean, it feels like that massive food supply would completely rewrite the rulebook for their fate map, wouldn't it?
Absolutely. You've hit on a key point. This is where evolution really shows its ingenious adaptability. The bird blastula is radically different.
Oh, so?
Well, it's not a hollow ball like in Amphioxus or even amphibians. Instead, it's a two-layered cap of cells, almost like a tiny flat disc sitting right on top of that huge, dense yolk mass.
Okay, a cap on yolk.
Right, and this structural difference forces a fundamentally reorganized fate map. The top layer of this cap, called the epiblast, is astonishingly versatile. It's responsible for forming a huge range of things.
Like what?
Get this. The extraembryonic ectoderm, those are membranes outside the actual embryo, as well as the embryo's own epidermis. Its neural plate, part of the notochord, all of the non-notochordal mesoderm, the yolk sac lining, and even the gut endoderm.
Whoa, wait, so one layer does almost everything.
Pretty much. The epiblast is the major player. Below it is the hypoblast. This lower layer contributes to the remaining endoderm and the rest of the notochord.
Okay.
But here's the really striking thing. In birds, the epiblast and hypoblast are not the direct equivalents of the ectoderm and endoderm we saw in Amphioxus or amphibians. It's a different setup,
So it's a whole different strategy.
a completely different evolutionary strategy for organizing the early embryo. Here, a single layer, the epiblast, essentially holds the potential for most of the primary embryonic structures. It really underscores how diverse life's initial architectural plans can be.
Okay, so we've peered into the embryo's future with these incredible fate maps. We know where things are supposed to end up. But knowing the destination is only half the story, right? The real magic, that breathtaking choreography you mentioned, happens in the actual dynamic dance of gastrulation itself. How do the cells do it? Let's maybe start with that relatively simpler Amphioxus again.
Good place to start. So remember, Amphioxus is isolysical.
Meaning a small amount of yolk, evenly spread.
Exactly. That minimal, evenly distributed yolk means there's nothing really hindering cell movements. So gastrulation there occurs primarily by a process called invagination.
Invagination, like
Right.
pushing a finger into a soft ball.
That's a great analogy. What happens is the vegetal area of the embryo, the bottom part, it first flattens out a bit, and then it literally tucks or folds inward. It invaginates. This transforms the embryo into something that looks like a tiny dimpled sphere,
Right.
or like your punched-in ball.
Okay.
As this inward folding, this invagination proceeds, the outer portion of the embryo naturally becomes the ectoderm. That's the prospective epidermis and neural system. The inner tucked-in cup, meanwhile, becomes the endoderm, the future gut, and its associated bits. And extending from the rim of this invagination, sort of rolling over the edge into the cup, we find the mesoderm prospective notochord and the rest. So at this stage, the Amphioxus gastrula has its primitive gut, the archenteron, and an opening to the outside called the blastopore. It's a beautifully simple, direct process that sets up the basic body plan. Very elegant.
Very elegant indeed. Okay, but that distinction you made earlier about amphibian eggs, that heavier yolk concentrated at the bottom.
The vegetal pole.
Right, that isn't just a nutritional detail, is it? It feels like that would completely mess up that simple invagination. It must make gastrulation way more complicated than in Amphioxus.
It absolutely
Yeah.
does. You're spot on. Amphibian eggs are moderately telolethal. That concentrated yolk in the vegetal region, it doesn't just slow down cell division there.
Uh-huh.
It crucially prevents that simple, direct invagination we just saw in Amphioxus. The cells just can't fold inward like that.
So how do they get inside then?
Instead, amphibian cells achieve gastrulation by migrating actively. They move from the exterior to the interior through a kind of groove that forms on the surface. That thick, yolky vegetal region simply refuses to be punched in. It's too bulky.
Right, so they have to kind of crawl over the edge.
Sort of, yeah. From a surface view, the events are quite a spectacle. First, a small slit, the blastopore, appears just below the embryo's equator. Cells from the surface then begin to actively move inside through this opening. This initial inward migration creates a region called the dorsal lip of the blastopore.
The dorsal lip, okay.
And interestingly, this forms right at the site of the gray crescent, which is a really important landmark on the early amphibian embryo.
Ah, okay.
As gastrulation continues, the blastopore doesn't stay a slit. It elongates, changing shape from a crescent to a semicircle and eventually forming a complete circle.
A circle, how?
It's driven by a continuous inward flow of cells from the surface. First, cells from the dorsal area move in, then the cells from the lateral regions, and finally the ventral cells join the party, completing that circular blastopore.
Wow.
And once it's fully formed, the center of this circular opening is often visibly filled with large, yolky endoderm cells. This creates that very distinctive feature you see in textbooks, the yolk plug.
The yolk plug. Right, so it's like the embryo is literally wearing its lunch on its sleeve, or rather its gut, for a little while.
Uh-huh. Yeah, that's one way to put it. It sits there plugging the hole for a time.
But what's the engine behind all this movement? You painted such a vivid picture of these cells literally moving in. Are they being pushed, pulled? Do we know how these complex movements are orchestrated?
That's the million-dollar question, isn't it? The precise mechanics. We observed that cells right at the dorsal lip region undergo these fascinating shape changes. They sort of elongate, contract, becoming almost bottle-shaped.
Bottle-shaped.
Yeah, it's reminiscent of what happens in sea urchin gastrulation, actually. This shape change seems to play a big role, actively pulling those cells inward and likely pulling attached neighboring cells along with them.
Okay, so they're actively involved.
Very actively involved. Now, the precise interplay of all the forces pushing, pulling cell-to-cell communication isn't fully mapped out yet. But we do know these dorsal lip cells are remarkably determined.
Determined? How so?
Well, experiments have shown that even if you surgically remove the dorsal lip and transplant to a completely different part of the embryo, those cells will still try to migrate inward. It's as if they've received their marching orders, their fate is set, and they're just going to follow those instructions regardless of their new location. It's incredible intrinsic programming.
That is incredible. Okay, so that's the surface view and the cell behavior. What about inside, if we took a slice through?
Right. Looking at cross-section gives another perspective. The cells destined to form the notochord, remember that supporting rod? They migrate from the dorsal surface, moving over that active dorsal lip, through the blastopore, and then they come to rest inside, just below where the neural tube will form. They end up forming the roof of the developing archenteron, the primitive gut. Meanwhile, on the outside, the ectoderm, which would be called the epidermis and the nervous system, keeps expanding, spreading over the entire embryo's surface like a blanket covering everything up.
Got it. Covering the migrating cells?
Exactly. And as the blastopore completes its circle, becoming round, that notochordal mesoderm continues to stream in dorsally, while the non-notochordal mesoderm flows in laterally and ventrally, filling out that crucial middle layer. Finally, the whole blastopore rim contracts, like a drawstring purse, pulling that visible yolk plug, those yolky endoderm cells, completely inside. And poof, it disappears from the surface.
Wow. It's just a remarkable self-organizing cascade of events.
It truly is. Complex choreography.
So we've seen the elegant invagination of Amphioxus, this more complex, yolk-driven surface migration in amphibians. Now, circling back to birds, knowing they have that massive yolk and that totally different blueprint, the epiblast, hypoblast cap, how does their gastrulation unfold? It must be an entirely different dance, right?
Oh, absolutely. Completely different. With that huge yolk mass, birds obviously cannot perform a simple invagination like Amphioxus. Nor do they engage in that kind of large-scale surface rolling or migration over a lip like we just described for amphibians. The geometry is all wrong for that.
So what's their trick?
Instead, bird gastrulation relies primarily on a process called ingression. And it centers around a very distinct structure that forms on the epiblast. It's called the primitive streak.
The primitive streak, I've heard of that.
Yes, it's fundamental. This primitive streak, which forms as a thickening along the embryo's midline on the epiblast surface, acts almost like a trough or a funnel.
A funnel?
Yeah. Epiblast cells from the surface converge towards this streak. They move towards it, then they detach from the epiblast layer, change shape, and migrate inward through the streak, down into the space between the epiblast and hypoblast.
So they dive through it.
Exactly. Once inside, these ingressing cells spread out laterally and forward to form the mesoderm layer and also contribute to the definitive endoderm layer, displacing the hypoblast in some areas. So it's less like a punch-in or a surface roll and more like a directed, continuous diving in or streaming in from the surface into the interior. It's a highly efficient way to establish those foundational layers in an embryo that's basically flat on top of a giant yolk.
That makes sense. A totally different solution for a different starting condition.
Precisely. And that really completes the comparative picture, reinforcing just how diverse these fundamental developmental strategies can be.
So wrapping this up, what does this all mean for you listening? We've journeyed from a simple ball of cells to this intricately layered blueprint of an organism. We've tracked these incredible cell movements that literally lay the foundation for everything to come. We've seen the elegant invagination of Amphioxus, the yoke-driven surface migration in amphibians, and this unique ingression through the primitive streak in bird embryos. Each one is really a testament to nature's diverse and, frankly, ingenious solutions for a fundamental biological challenge.
Hmm.
How do you transform a simple collection of cells into the complex, multi-layered beginning of a new life?
Yeah, and what becomes really clear is that while the goal of forming a basic body plan with distinct layers is universal across these animals,
Great.
the evolutionary paths to get there are wonderfully diverse. These initial gastrulation strategies, they're meticulously tailored to the embryo's environment, its resources like yolk.
Yeah.
They aren't just fleeting early events.
To the lasting effects.
They have deep and lasting effects. They fundamentally influence every subsequent developmental step. They shape the unique anatomy and physiology of each organism, from a tiny, simple Amphioxus to a complex bird, and ultimately to creatures like us. It really encourages us, I think, to appreciate the hidden complexities, the beautiful choreography that underpins all life. And maybe it leaves us wondering, how do these very different foundational beginnings continue to reverberate through an organism's entire existence, shaping the final form?
A fascinating question to ponder. How those first steps echo all the way through.